hypermethylated that it essentially turns off gene expression. This has been known to be deregulated in cancer, creating either hypermethylation or hypomethylation. It's been increasingly recognized that this is an important process in oncology, and there have been numerous examples of genes that are being hyper or hypomethylated, and that this is an alternative mechanism compared to copy number alterations and DNA mutations. For example, MGMT methylation, hypermethylation in glioblastoma, and BRCA1 hypermethylation in ovarian and breast cancer. Also, there have been in the literature global methylation patterns discovered in multiple cancers, and this is often called a SIMP phenotype or CpG island methylator phenotype, and this phenotype is characterized by hypermethylation. It has been shown in colorectal cancer, where it's defined by BRAF mutations. It has also been shown in gliomas, where it's defined by EDH1 mutations, and also in AML, where it's caused by IDH1, IDH2, or 2 mutations. But studying methylation, there has been a lack of formal statistical methods to identify hyper and hypomethylation in cancer. Also, we need to take into account that we need to know the normal state, because we don't really know if a gene is supposed to be hyper or hypomethylated. Also, we decided to focus only on methylation changes that are functional. So we want to only focus on methylation changes that have an effect on transcription or on gene expression. Previous methods that are looking at this typically do not take into account gene expression. So I'm going to compare our algorithm with 3 methods, IMA, COH, CAP, and MinPhi. And all 3 methods are characterized by the fact that these are essentially statistical tests that look at cancer versus normal. Also, MinPhi, for example, this only focuses on hypermethylation. So we developed MethylMix, which is a new algorithm to identify hyper and hypomethylated genes. We wanted to take into account the heterogeneity in the data, and essentially the model behind MethylMix is a mixture of beta distributions. This takes into account this heterogeneity by modeling subpopulations in cancer cohorts. Also, we compare with normal tissue, and finally we incorporate matched gene expression data. So this is an example of I don't know if there's a pointer. So this is an example of a histogram of methylation values based on a microarray platform that is used in TCGA. So on the x-axis, you can see a range of 0 to 1, where 0 means that the gene is completely unmethylated, where 1 means that the gene is completely methylated. So these are essentially proportions. And in this case, you can see that the dominant methylation is around 20% methylation. So what we do is we model this as a distribution of beta mixture models. And we use the Bayesian information criterion score to identify the number of modes that are in the data. And as long as the Bayesian information criterion score improves, we keep adding modes. For example, in this case, we identified 2 modes, where one models the dominant methylation pattern, and the second mode has a higher methylation status. Next, we compare these modes with the normal methylation status, and here you can see on the top of the green mode the 95% confidence interval of the methylation of this gene for these samples. And you can see that the variance of normal methylation is much lower than in cancer. So what we have identified here is essentially that the green mode is the normal mode, whereas the red mode is essentially hypermethylation. And in this case, this was an ovarian cancer data set from the TCGA, and this gene was a BRCA1. So essentially, we have now identified BRCA1 hypermethylation in ovarian cancer using a statistical method where no manual thresholds have to be made. And finally, we also integrate gene expression data, and we focus only on inverse correlations between gene expression and methylation status. So we really only want to see these relationships where we see a differential methylation pattern which has an effect on gene expression. So to summarize, so we have the raw data. We model them using a beta mixture model, and this allows us to identify methylation states. The normal state allows us to annotate the states with either hypo, or normal variation. This gives us essentially differential methylation, and finally we integrate gene expression data. And this gives us essentially differential and functional methylation states for each gene. And so this whole process is repeated for each CpG site or gene. So we used our algorithm MethylMix and we applied it on the Cancer Genome Atlas data. I'm sure I don't need to introduce this data set. So we used about 12 cancer sites, and you can see the number of samples for each cancer site, totaling over 4,000 cases. So first what we did is we applied MethylMix on each cancer site separately. And this identified between 234 and 798 hypermethylated genes, where the most hypermethylated genes were identified in breast cancer. Next, we also identified hypomethylated genes, although there were less, between 74 and 251. And we also identified dual genes because we don't limit the number of modes that are being modeled. So we also can have a situation where a gene can switch from a hypermethylation status or a tumor suppressor context to an oncogene context. And overall, you can see the average that we identified across all these tumors. So first, what we did is we also looked at the top hypermethylated genes. So which genes are being hypermethylated in many different cancer sites. And we found a number of genes that are methylated in at least, hypermethylated in at least 10 cancer sites. So overall, we found about 266 of these pan-cancer hypermethylated genes, and this was defined as at least 5 cancer sites. So just to give you an example, the top gene, which was hypermethylated in 10 sites, this is TMM25. It hasn't been well studied. It has been shown to be hypermethylated and downregulated in colorectal cancer, which is also what we see in our analysis because it was 100% hypermethylated. It's also been shown to be a favorable prognostic marker in breast cancer, and we see 6% hypermethylation. Similarly, we did the same analysis for the top hypomethylated genes. And here we found a little bit less genes. So the top genes were hypomethylated in 8 cancer sites. And overall, we found 42 pan-cancer hypomethylated genes in at least 5 sites. Again, focusing on the top gene, A4, it's hypomethylated in 9 cancers, which is prevalence. It's a target for immunotherapy. It's known to promote cancer cell growth, so which would correspond to a hypomethylation status. It's also a therapeutic target in breast cancer. And more importantly, the MHA family is currently being tested in phase 3 trials in lung cancer. So next what we did is we wanted to compare our methods with IMA, COH, CAP, and MinFi. So what is the difference between the genes that are identified by MethylMix and these 3 other methods? So first, here are again the numbers of hyper and hypomethylated genes that are being identified by MethylMix. And here are the numbers for IMA, COH, CAP, and MinFi. And what you can see is that these other methods essentially identify much more genes. So why is this? So first, we looked at which genes are exclusively identified by these other methods. And what we found is that very few of these genes are functional. So these methods do not integrate gene expression data. So this explains the difference between MethylMix and other methods. Is we are more stringent because we only want to focus on functional differential methylation. So only on methylation changes that have an effect on gene expression. Next, what is special about the genes that MethylMix exclusively identifies? So what are the genes that MethylMix identifies and are not identified by the other methods? So what we found is, and I'm only going to show the results for IMA. No, I'm going to show the results for all 3 algorithms, is that the other algorithms cannot deal with the heterogeneity in the data. So MethylMix identifies more differential methylation for methylation events that happen in less than 15% of the cohort. So these other methods just do not take this into account. And this is the same for both hypermethylation and hypomethylation. And for MinFi, MinFi did not identify hypomethylation. And finally, we wanted to do a sort of orthogonal analysis to compare how each of the 4 methods essentially is able to identify cancer driver genes by using some independent orthogonal information. And to do this, we used the cancer stage. So we looked first at genes whose expression is correlated with increasing or decreasing cancer stage. For some cancers, cancer stage was not available, and only for 4 tumor types, we were able to identify enough cancer stage driver genes. So in these 4 cancers, breast cancer, renal cell carcinoma, lung adenocarcinoma, and endometrial cancers, we were able to identify enough cancer driver genes using this independent information. And we split them up in oncogenes and tumor suppressor genes, and here you can see the numbers for each of these cancer sites. So what we did next is we looked at how well MethylMix and these other algorithms can essentially identify genes that overlap with this orthogonal information. And we found that MethylMix essentially was more enriched in these type of genes. For example, for the KIRK or renal cell carcinoma, MethylMix was enriched with over 48% cancer stage drivers, whereas the IMA method identified much less of these genes, only 30%. Next, we also wanted to use these methylation data analysis for subgrouping. To do this, we introduced a new statistic, which is called the DM value, and essentially this reflects either the hyper or hypomethylation state. So the DM value is defined as a difference between the methylation mode and the normal. So for hypermethylation, this creates essentially a DM value bigger than 0, and for hypomethylation, smaller than 0. Then we use consensus clustering on the DM values to define subtypes. And we used default parameters for consensus clustering. And in this way, we essentially identified subgroups in each cancer site. So first, we focused on the known SIMB subgroups, and if we could rediscover these groups using MethylMix genes. Here you can see the 3 main SIMB cancer types: colorectal, glioblastoma, and AML. And what I'm showing you is a consensus, which is essentially the samples versus the samples. And dark blue values mean that the samples cluster together in the same cluster in each of the subsamples, where white means that the samples never cluster together. And what we observed is that we essentially are able to identify the SIMB subtypes. So this is shown on the bottom, where the hyper or hypomethylation is visualized, with red meaning hypermethylation and blue is hypomethylation. So you can see that in each of these cancer sites, there's one subgroup which has a lot of hypermethylation. And this was then verified with no SIMB annotations for these 3 tumor types. Next, what we did is we wanted to identify if these DM values really improve the clustering. If we are using only MethylMix genes, do we really find a better clustering? And what we observed was that essentially these DM values denoise the methylation data compared to raw methylation values. And this is shown here. So what I'm showing you here is the intra and inter-cluster consensus. So the intra-cluster consensus, we want to be as high as possible to 100%. We want the samples within each cluster to be as similar to each other. Whereas the comparison between consensus between clusters should be as low to 0% as possible. So here is, I'm showing you the values for the DM value clustering. And on the right is using the same genes, so the MethylMix genes, but using the raw beta values. And what you can see is that consensus was almost in each case better with the DM value. So essentially, we are denoising the data. Finally, of course, besides just looking at known groups, we wanted to look at novel subgroups. And I'm going to show you results of 2 cancer sites. So this is the renal cell carcinoma data set where we identified 5 clusters. And we also correlated these clusters with survival, and we found significant correlations. And I also want to draw your attention to subgroup 5, which is the subgroup on the right, which essentially seems to be a normal-like subtype because it has very few or fewer hyper and hypomethylation genes. Another example is the head and neck squamous carcinoma, where we again identified 5 subtypes. And in this case, we correlate these subtypes with known mutations. Again, subgroup 5 seems to be devoid of known mutations, so TP53, NOTCH1, and CASP8 mutations, whereas subgroup 2 seems to be caused by a mutation in a gene, which is a methyltransferase gene, and this is causing the hypomethylation that you can see for subgroup 2 at the bottom. And we also identified that these groups were correlated with survival, especially subgroup 5, which is void of canonical mutations, seemed to have a very good prognosis. Finally, we decided also to do a pan-cancer analysis where we essentially combined all cancers and normals and ran MethylMix on all cancers combined. But to do this, we needed to deal with the normal genes because the normal methylation is very known to be a cell type marker. So we needed to treat we needed to look for normal genes that essentially are unimodal, because otherwise we have a very heterogeneous normal to compare with. So to do this, we ran MethylMix first on the normals to identify these unimodal genes, and then we did a pan-cancer analysis only on the unimodal genes. And then we did again consensus clustering to identify essentially pan-cancer methylation-driven subtypes. And we found 10 subtypes. 4 of these clusters virtually contain all and only samples from one tissue. These are the glioblastoma subtype, which is very well known to be a very different tissue. The colorectal subtype, the leukemia subtype, and also the renal cell carcinoma. So all these 4 clusters were very well very tissue specific. Next, we also found 2 clusters which contain only samples, subset of samples of one tissue. So these are all the breast cancers, but only the non-basal breast cancers. And similarly, for the endometrial tumors, these are all the non-serous endometrial tumors. And finally, there are 4 clusters which are of mixed tissue. Cluster 2, this is cluster, the serous ovarian cancer cluster, which also contains serous endometrial samples. So this means that essentially serous endometrial cancers are more similar to ovarian cancers than other endometrial cancers. There's also a mixed lung cluster. This cluster contains lung adenocarcinoma, lung squamous carcinoma, but also basal breast cancers and some head and neck squamous carcinoma. And there's also a second mixed lung cluster, which is more rich with lung adenocarcinoma, but also contains other tumor types. And then finally, there's a squamous cluster, which contains squamous tumors from various tissues. So these are the 4 clusters that essentially contain mixed tissues. And we wanted to understand these clusters better, so we used gene set enrichment analysis to identify which pathways are overrepresented in each of these clusters. And for the 2 subclusters, we essentially identified that the lung mix cluster 1 is enriched in the cynic and integrin pathway, whereas the lung cluster mix 2 is enriched in the FOXA immune response pathway. So this, especially the second cluster, was very interesting because I have to stress that we only used exclusive enrichment. So these are pathways that are exclusively enriched in one clusters versus all the rest. Finally, we compared our analysis with pan-cancer subgrouping based on other types of data, such as mutation data. And essentially, the conclusion is that none of the other data types essentially pick up these unique clusters. So to summarize, we developed MethylMix to identify hyper and hypomethylated genes. We used them to an pan-cancer application. We used the M values to cluster and identify new subgroups. And finally, we did a pan-cancer clustering. These are the people I have to acknowledge for this work. And MethylMix is available as an R package in a bioconductor. Thank you for your attention. I'll be happy to answer questions.
So we have 2 minutes for questions. Any question, please use the microphone at each side. Okay. Any questions?
Hey. Does it work with all types of DNA methylation data, or RBS, whole genome bisulfate, as well as 450K? Or?
Can you repeat that, please? Sorry.
What platforms can you use it with?
So for now, at this moment, this is based on the Illumina microarray platform. So we're using both the 27K and 450K data. We're thinking about extensions to bisulfite sequencing, but due to the mixture modeling approach, we need actually a lot of data. So the cohorts for sequencing data are not at a decent size yet that we can use this model.
So would 22 samples be enough?
That's very borderline, yeah. I think we need, typically the smallest cohorts that we use on is about 100 cases.
You mentioned that you start with the filtering the genes that you use to analyze the data. So I was wondering whether that pre-filtering may have any impact on the enrichment analysis that you perform later on.
What do you mean exactly with the
You're
pre-filter?
saying that you look at the only at the genes that you have unimodal methylation, so that presumably it's not all the genes. So I was wondering how that may relate or influence your enrichment analysis.
So for the enrichment analysis, we just we did not use those filters. So we went back to the gene expression data, and it's and we only focus on upregulation.
Okay, thank you.
Yeah.
Okay, so the second talk is MIME cover, integrated analysis of